Hello everyone, thank you for joining another session of the ISCB Student Council webinars. Sorry for the delay, we had some technical problems, but now we are ready. Today, Sofia Celi will present about the assembly of an scorpion genome. So please start.
Thank you, Katrina. Hello, everyone. My name is Sofia Seli. I'm a master's student at the Faculty of Medicine and Pharmacy at Rabat. And currently, I'm doing my internship in the University Mohammed VI of Health Sciences in Casablanca and the HPC Marwan department at the CNRC in Rabat under the supervision of Professor Hassan Ghazal and Professor Shakib Nisar. So I'll start by introducing the Androctonus mauritanicus scorpion, then the importance of the venom production and signaling pathway, the aim of the study, biosample and sequencing, the two assembly approaches and how we estimated the genome size, our assembly outputs, conclusion, and perspectives. So the Androctonus mauritanicus, or also known as the Fattail scorpion or Fattail scorpion, is the common name given to scorpions of the genus Androctonus, which are endemic to Morocco. These scorpion has a chromosome number equal to 24 and is one of the most dangerous scorpion species in Morocco, as the national strategy against scorpion stings reported that approximately 25,000 stung patients are recorded every year in Morocco. So the venom is also a rich source of biologically active peptides constituted by mucopolysaccharides, hyaluronidase, phospholipase, serotonin, histamine, enzyme inhibitors, and proteins, namely neurotoxins peptides. And they also have the non-disulfide bridge peptides that constitute an important group displaying multifunctional activities. For instance, the moriporin, which is a peptide composed of 48 residues that may offer a novel therapeutic strategy in the treatment of prostate cancer, considering its significant cytotoxic potency against prostate cancer cells and low toxicity to non-tumor origenic cells. For the venom signaling pathway, as we can see here, we have the venom neurotoxins, which can be either the alpha toxins or the beta ones. They affect the parasympathetic nervous system or the sympathetic nervous one through the sodium channels, and thus by secreting the acetylcholine or the catecholamines that will respectively bind to cholinergic receptors and the adrenergic receptors, which will result in cardiovascular disorder and then an acute pulmonary edema, which may cause death. So one of the scorpion venom has a therapeutical potential. For example, in the inhibition of growth of cancer cells. One is the blockage of ion channel in cancer cells, the inhibition of metastasis and invasion of cancer cells, and the activation of intracellular pathways leading to apoptosis in cells. So with that being said, the sequencing and the assembly of the Androctonus mauritanicus genome is very important so that we could understand these pathways and maybe make them artificially for therapeutical purposes. So here's the general strategy workflow. So far we have done the sequencing and the assembly. The next steps are the structural annotation and the functional ones. So coming to the biosample and the sequencing, the biosample was an adult black scorpion And the DNA was extracted from the leg tissue with the Qiagen Fast DNA kit. Then library preparation was performed with the Illumina TrueSeq, and sequencing was done using the Illumina HiSeq Extend, which generated 107 million reads of 150 base and a total size of 32.3 gigabase with a coverage of 120 bits. So there are two major approaches of assembly: the mapping alignment, or the alignment assembly, or the de novo assembly, and this relies on whether a reference genome sequence exists or not. So for the mapping assembly, you just assemble your sequence reads into contigs by mapping the reads against a reference genome sequence. While the de novo assembly, we assemble our sequence reads without using any kind of template or reference genome sequence, so it's kind of going from scratch. So for a moment, back to the mapping assembly. As we can see here in this blue line, we have the reference genome sequence, which is a known one. And here we have our row reads, which are going to be mapped to this reference genome sequence. For the assembly method we used the de novo assembly for many reasons. One of them is it's better for unknown or less characterized organisms, and it does not force your assembly to what is already known, allowing discovery of the novel gene genetic configurations. For the disadvantages, well, it's more computationally demanding, and it is not as good when looking for tiny differences in well-known genomes like the human one. So the de novo assembly looks like this. Here we have our chromosome, which originally we took the genome. Here we have our row reads that were sequenced. When we put them together, we get contiguous sequences that are referred to as kinetics. Then when you put these kinetics together, you get something called scaffolds. Then by this, we go back to our chromosome. For the assembly algorithms, there are three known. There are three ones: the de Bruijn graph, the string graph, and the overlap graph. The oftenly used one are the de Bruijn graph and the overlap graph. For the de Bruijn graph, it is usually suited for short reads and uses chimers. But for the overlap graph, it is mostly used for longer reads. So here we have some genome assembly softwares that are classified according to the assembly algorithms. For us, we used Spadis, which is a de Bruijn graph-based assembler and is well suited for short reads produced by the Illumina sequencer, for example, and uses multi-chimers and can also choose chimers automatically based on read lengths. Some of the assembly challenges remain, which are common in the eukaryotic organisms, such as the repeat sequences resulting in copy loss or gain in our assembly, uneven sequencing depth that impacts the genome assembly. We have also heterozygosity and the genome size in arthropods that goes from 100 megabase to 10 gigabase. So here I summarize some assembly experiences with arthropods that have already been done. So there are more than 17,000 bioprojects of arthropods, including 1,108 genome already assembled, and there are projects including the I5K initiative, which aims to sequence 5,000 arthropod genomes, and the Iridian Genomes that have already 148 bioprojects of insects, including 49 already assembled. So before the assembly, we run some chimer frequency analysis to estimate the genome size with using the DSK tool. So here we have the chimer frequency plot from which we estimated the genome size, which is equal to 1.02 gigabase. Coming to our assembly output, we have here our scorpion genome assembled with Spadis, and here are two other scorpions that were done. So the estimated genome, as you can see here, is clear, is very near to the other one, done with velvet. The N50 is for us is higher. The number of scaffolds also is higher. The total size we found 1.5 gigabase, which is 50% higher than the estimated genome size. So in order to evaluate if our genome assembly is good enough to construct genes, we looked into the, we have done a benchmarking with the autologous analysis in the arthropod database. So with among these 1,030 genes we searched, we found that 44.3% of these in our assembly were completed autologous. Among them, 20.7% were complete and single copies, 23.6% were complete and duplicated, 10.3% of fragmented autologous, and 45.4% were missing autologous. So to conclude, our genome size assembly was 15% higher than the estimated genome, and about 45% of the arthropods autologous were missing. The assembly didn't reach chromosomes construction. Last but not, for our future visions here, we would like to annotate the genome using the NCBI annotation pipeline, doing a comparative genome with study with other Scorpio genomes, and of course do transcriptomics analysis for accurate annotation, and study the Venom pathways analysis even more, so that we can have a deeper understanding and open doors for therapeutical purposes for cancer. So last but not least, I would like to thank Stacy Thoreau from the Iridian Genomes, Salsabil Hamdi from the Pasteur Institute at Casablanca, and Tatiana Tatusova from the NCBI, Bushra Rahim, and Professor Ridwan Marouche from the Marwan department at the CNRC at Rabat. So thank you for your attention.
Thank you so much for your talk, Sophia. It was very interesting. Now we can wait for any questions from the audience. And meanwhile, I want to remind everyone that you can visit our social networks, Facebook and Twitter, to learn about other webinars and how to participate in the future if you are interested. There is a question. It says, why did you choose these species of scorpions to work on?
Well, because it is very distributed in Morocco. And as I already said, the 25,000 skunk patients were reported every year in Morocco from this species.
I also have a question. What are there any genes that interest you aside from the ones related to venom in this scorpion? What are you looking for, or what will you be looking for when doing the annotation? Is there anything special you want to focus on?
Well, its venom is very rich in many biologically active compounds. So maybe we could identify a pathway for a certain protein that may be involved in therapeutics. which is a good thing.
Thank you. There is another question. Why the assembly size is higher than the estimated genome size?
Oh, for that, I think that it is due to the chimer frequency analysis. So they are not too accurate. So we don't really rely on them. It is best to see other studies and compare the assemblies of other scorpions, like I have done in the table. And this is, of course, just a first try with Spadis. I will use other tools in the future.
Sorry if I missed this and you explained it, but this software you used is usually used for bacterial genomes, for smaller genomes. Why did you choose it for this more complex genome?
Well, because we have short reads, and it is well suitable since it uses the brain graph, and it also uses multi-chimers, which is also good. And as I said in the slide of Spadis, I said that it automatically chooses chimers depending on the read length.
Mm-hmm. Thank you. So I think I think there are no more questions. Thank you so much again. I'm sorry for the problems we had when starting. So yeah, have
Thank you,
a good
Jana.
evening or day. Goodbye.